Hey, what's up, everybody? I just want to walk you through this mitosis virtual lab just to help you troubleshoot any issues that might pop up. So the whole point of this lab is to kind of like through your own investigations try to figure out how long each phase of mitosis lasts in a cell. So a way that you could do this, and we would do this in real life, is we would take a sample from a cell of cells. So say like from your cheeks and put that on you could put like your cheek cells on a slide. Should usually do that one. Or like you take a little tiny bit of onion uh, from like the root where it's growing, because there's a ton of new cells growing there. There's also a ton of new cells being or dividing in your cheeks too, right? Because that's like epithelial cells, like the cells on the inner lining of your mouth get shed and rebuilt all the time. So anyway, there's a lot of mitosis happening there, and in the root tip of an onion. So that's why we picked the onion root tip for this example. And then you're going to be because we can't do this in person, you're going to be looking at an online version of a slide and under a microscope at these cells, at these onion root tip cells. And so when you click this link, and when you click this link, it'll take you here to Arizona.edu. And this is like one of the classic virtual labs that's been around for a while. So imagine this is the slide, this little box here that you're going to take a closer look at in a second. Okay. And your goal is to figure out how many of these cells are in each of the phases, right? Interphase, prophase, metaphase. And I can kind of tell just from here that one looks like it's in telophase, right? Like the chromosomes are all the way on the opposite sides of the cell. It looks like it might be starting to divide. And in plants, the cytokinesis happens a little bit differently because it's like it builds a new cell wall in between the cell versus like pinching off. Anyway, okay, so you read this stuff. Read. Click next. Read. It's a great overview of the phases and what they look like and what's happening in those phases. Next. And then you're going to have a copy of this, which you already do on this document. You're welcome to make a copy with pencil and paper. And then click next. So, and it's going to actually keep track of the data for you. So you could just copy the data down after you're all done. But you're going to get like one little cell from that slide over here on the left. So what phase does this cell look like it's in? To me, that looks like metaphase because all the chromosomes are lined up in the middle and being ready to be separated. Oh yeah, correct. So it puts it in the metaphase category. Okay, this one, I'm gonna just intentionally get it wrong so we can see what happens. But let's say I chose telophase. Ooh, not so much. This cell has a prominent nucleolus, the dark spot in the nucleus, which is not a feature of cells in telophase. So let's try again. Let's put it in the correct location of interphase. Oh yeah, check it out, and put it in the interphase box. Now I'm going to get one more interphase, and I'm going to need the first three answers, but it's just a good way to explain what's going on. And it's going to also put it in interphase. So like, look, I'm going to end up with 2, and then I might end up with more as I go along. So go through all of the cells that they give you to identify, and eventually you'll have this data table filled out with the number of cells in each of these phases, total of 36 cells, and the percentage out of 100%. And the whole reason we're doing this is to get a sense of like, what does real science look like if we're trying to figure out how long a cell is in each of these phases. We can't I guess we could watch a cell divide. Actually, I have. It's really cool, and you could time it, but that's really time consuming, and you would need like a lot of different samples of it. And so, a great way to do it is take a snapshot of a bunch of cells, and depending on how many are in interphase or telophase, you can kind of guess like how long do cells spend in that phase based on that data. And so I'm not interested necessarily in you guys memorizing these words and having them in your brain forever, but more so about the process of how science is conducted. And then also just a little bit of like cellular biology and a little bit of microscopy, which is how to use a microscope, which I know we're not quite getting in this scenario because of the virtual stuff. But and a side feature of this assignment is that you also learn how to create a graph in Google Sheets. / use Google Sheets, which is a hugely transferable skill. Knowing how to use a spreadsheet and how to navigate your way around a spreadsheet is really important in today's world. So hopefully that will also not be super boring, but you'll see the upside in why I'm having you guys do this. And there are some questions to answer that dig a little bit deeper, make you think about it in new contexts, right, for critical thinking, and bring a little bit more meaning to the work that you've done. So that's how you do the mitosis lab. I'm sure you've skipped around this video or have stopped watching by this point because it's a little bit slow and rambly, but hopefully some people found it helpful.